see which camera. Okay, well, we're doing this camera. That's good. Let's share my screen. All right. Go back to bacteria stinks. Can't wait to get rid of it. I get like a couple weeks at the end of the semester where I don't have to have as much bacteria. I'm looking forward to it. Let's see. I'll open chat. You open this up too.
Do you have any big plans for Thanksgiving break?
My husband and I are having my parents and his parents over. Like it's like my sister and like her husband's family, they live in Texas, and so like we try to alternate like when we're doing Thanksgiving and where we're doing Christmas and stuff. Thank you very much. Thank you very much. But so it's supposed to be our off year, but I'm hoping this parent is on fire. I'm not doing it. I'm living in. So I would be not doing that. And then my my parents, again, it's just me and my sister, and so my sister is doing it in Dallas by themselves. It's almost like, they live in Columbia, so, it's a drive for them. They say that it's gonna be. Yeah, so if it's bad, they might not come back. Anyways, the point is, we're, and so, yes, so it's not terrible. It's not like, what terrible movies, like shows like you see on, TLC. But, it is bad enough where like, if people go work, it's like a huge stress to try to get it organized enough for people to be over. It's gotten to a point where like, almost a little bit until it just kind of, and we don't worry about it. We actually have to keep on over and whatever, so it's less stress on her. They're old enough, like I'm not gonna do that. So, but yes, so we're gonna try to smoke a duck. We have never smoked anything. My husband, it's been coming, and I'm like, I said we were gonna smoke. We were gonna smoke a turkey, yeah, but now it's just 6 of us, and we're like, probably don't need a turkey. So, I was like, how about the roast chicken or something? So we could do that, but now I'm like, we'll roast it. It'll be fine. You can put it on the smoke it. That's hot in the smoker for his anniversary, for our 10-year anniversary. And he's never smoked anything ever. And so I keep like being like, maybe smoke a chicken or smoke like something. Smoke something.
Starting beer.
Yes, and then you don't have to rely on this being there for Thanksgiving. So he keeps saying, yeah, this would be, and he's going to smoke a chicken. And I'm like, yeah, that's a great start. And so, but like, we're already at Wednesday. I'm like, when are you gonna?
Time's taken.
Yeah, and I don't mind roasting the duck. That's easy enough. We've done it before, so we can do that. But it's also like one of those things where I'm gonna try. It has the wood chips. We have like the box for the wood chips, and we have the charcoal. We have like everything ready to go. It's just a matter of actually.
Yeah. Getting nervous.
That's his one job though. He'll be whatever we end up doing with the duck. He'll be doing the duck, and I'm gonna make green bean casserole. And I do this thing where I make cranberry sauce from scratch. Cranberries, we make it with boil the cranberries with orange juice, and I squeeze the oranges fresh, and then it'll gelatinize itself. Not gelatin, obviously, but like the sugars in there can deal with all. It's more like a combo. And you actually do obviously because there's a very hard to work. Yes. Yeah, and it'll go really well. Which you guys have had that before. It is so good.
I think I tried once.
Yeah, it's very fatty, and it goes really well with fruit. Usually I will do, they call it duck vanilla orange or something like that. And so usually I've done that before in the past. But yeah, this should be.
I'll take that. I'll have to ask you about it after break.
Yeah, we'll see.
Or maybe I
I will say his parents are bringing a ham too. So we will have backup. Yes. What about you?
At least get to be home.
Yeah, what are you doing?
Our family lives out in Levy and Woodward, and so we'll leave Monday after the last.
Yeah.
Be out there till the next.
Yeah, that'll be nice. Get away, take a breath.
Yeah, the kids were wanting to leave Friday to go hunting longer. I was like, I'm not going back. Yeah, because they were loud.
Yeah, that's the thing about the lab. I don't. That was like the one place I needed to have space, and it like really. I tried.
No,
I tried to change the rules. I tried. They were like, no, let's just have to be in class. I know other professors get away with not having class on Monday, Tuesday, but I'm not here. So I'm not playing that fire. Like I want
We don't have live on.
yeah.
so it's not bad.
Campus will send out an announcement about Wednesday being closed. It has closed every year in the past. So I'm just leaning on that happening. We don't have any for Wednesday. In fact, my Tuesday night class has all agreed to take theirs on Monday night so that I don't have to stay. So, yeah.
That would be nice.
Yeah. So I just have Monday and then Tuesday morning. Good.
Oh, wait, no.
All right, it is 11:02. Let me see. We've got 12 people. I'm gonna go ahead and mark off who I know is already here. Julie Jackson. Catherine P.
Are you going to do a Sunday review before the final or no?
Yes, so we'll do the same thing as usual.
Monday or Sunday.
So we will have a Sunday review before the actual lecture final. So not before the lab final, right? This is it for the lab, right? But for the lecture final, we will have a Sunday review on Sunday before the actual final. And it will be a longer one. So it will be about an hour on, Kahoot can only make quizzes up to 100 questions. So I have two 100 question quizzes. So 200 questions this time to try to cover as many topics in as many ways as we can. So we'll do one of the quizzes for about an hour and then the other one for about an hour. It's roughly unit 1 and 2 and unit 3 and 4, but there's a lot of crossover on both of them. So we'll play one of the quizzes and then one of the other quizzes, and you'll get links to both of them too. So if you want to play them on your own time, you can. It's the same drill as always. Like, I'm going to let you guys play the test. I'll try to review things that we do bad on. I'll write up the notes on the things we did bad on and post those in the announcement. It should be the same other than that. It's a good question. Any other questions before we get started with the lab review? Okay. So we've had students going to the FITME. So far, did you guys find it helpful? You did? Okay.
I just wonder if like the lab.
Basics, sort of, yeah, the basics in the lab, like study sheet thing, and yeah, so good. So far, I've had, I'm not even kidding, like, I could do on my Tuesday morning class, you guys heard me do it before, not regularly, right, but they just had a couple of incidents lately. They don't perform as well, but, you know, yesterday, when we had class, and I was like, hey, you know, you guys, it's still on you, you can still do it, you can still do better, like, we can still save our grades, you can still do fine, we just gotta show up and do the work, right? And I called Sydney, and I was like, hey, you know, I'm going to bring by some more of these sheets because I only printed off like 10 at first. I want to bring by more of them. Have you had anybody show up? She's like, yeah, we actually have some students right now. So I went over there, and literally 7 of my students in my Tuesday morning class were all there doing group work with her. And I'm like, yes. And so you guys went. It's been helpful for people. If you're looking for a way to help you study and talk it through somebody that has reference that isn't me, and she's literally there for her. And you're already paying for it. Go and replace some back scores in the meantime, you might as well. So she has the forms there. If you guys go, so for example, if you go for an hour, 30 minutes will replace the normal exercise. So it can replace low scores or even zeros. It can be for extra credit or any normal assignment, not projects or exams, right? So just a reminder about that. And then you just bring the paper to me. If you get it done and I'm not in my office, just slide it under my door. So there might be times where I'm like in class with another class or something like that. Just slide it under my door if I'm not there or not in my office, or whatever. So FYI, just get those papers to me up to final exam day. You can hand me those papers. Another reminder about the final exam. You know, I'm pretty lenient about like when you submit things. If we submit it half a day late, I usually remove the late penalties. Maybe not the quizzes because they're kind of auto, but like your lab, we've had some issues with some students. Maybe our concept maps got submitted a little late. I usually remove the late penalties. It's only like a day or two. I'm pretty lenient with that. But once we get to the final exam, that's it, nothing else. So if you get the final exam, you're like, wow, I did bad. I'm going to try to submit these other things again. Nope. Final exam is the cutoff. Once you submit the final exam, that's your grade. On final exam day, so I don't mean the lab final, I mean the lecture final, the final. So once you get your grade out from the lecture final, again, I have to hand grade your things. I will make an announcement saying all grades are now final, and that's it. Okay. So once you submit your final, you can't submit anything else. And then I will notify you once your grade is finalized. Because I'll grade the final exam and then post the extra credit because I have to have the grade for the final posted before it'll count extra credit as extra. Otherwise, it counts as an assignment. And then people who didn't do it will get zeros on an assignment and it will bring down their grade. You can't have that. So I have to submit the extra credit after I grade everything. So hold your breath after you take your exams, okay? Like it's coming. I will probably have all of the exams graded that same day, but give me at least one day after that. And if for some reason you're still concerned about it, Eve, shoot me an email. Odds are though, I don't see that happening, like, unless I, got hit by a car and died or something. You know how, you know how I am. I'm going to try to, I go out of my way to try to get that stuff done for you guys. I don't get it, but this is what I do, is get stuff done. So, stay on top of that, watch your grade. It should be by, at the absolute latest, Tuesday night, that your grade will be finalized. And I will make an announcement about that as well. Pretty soon here, you're going to get emails about the course evaluation stuff too. When that opens up, if you submit a course evaluation, you can get half a point of extra credit. I mean, half a point, a percentage point added to your overall grade. So it's the last half a point that you can get for extra credit. It's our last extra credit opportunity. And it is just for doing the course evaluation. You don't need to show me your answers. I don't need you to give me a good score. Give me an honest, rate the course appropriately. You don't even have to answer the comment questions, but I would appreciate it. Like if you're giving me less than a full score, I would love to hear about why that is. Oh, there's too much homework. Oh, we didn't use the, textbook or whatever it is that you feel about the course. I want to hear that because that's usually how I base my changes on my course to feedback from you guys. So I do read those, but they are completely anonymous. Just a reminder, course evaluations are completely anonymous. I never see who posted what on those course evaluations. You just have to send me a screenshot saying that you completed, and I'll add in those points for you. Again, extra credit's not factored into your grade at the end of the semester, but they'll be there. And if you need help figuring out your extra credit, saying on me or whatever, and so you can estimate what your grade is going to be. It won't pop up in your what if calculators, like when you submit the grade on their grade book side. Makes sense? We all tracking that? Okay, so you'll get plenty of emails about the course evaluations. You'll get them from me. You'll get them from the university. And it'll be hard to miss. And now that they have the new system in place, I think it's easier to track that too. It's easier to follow that. So I know if you guys lose your link or you don't get your link in your email, because sometimes it goes to your junk mail, you can just email me and I have access to the link. So I can just send it to you if you can't find it. When those come out, you'll get plenty of reminders about it. I thought they would be out this week, but they aren't. So, okay. So, all right, let's do this. Let's get started. Any other questions though before we get going? Okay, that's fine. Stay as long as you can. All righty, so let's do this. And for my online people, I'll try to hold up the tubes to the camera. It might be small, so just wear that. We talk about some of the tubes. All right, so first things first, the lab final is going to be testing on the microscope function, aseptic technique, and isolation. Now, we will have, you guys have seen it. You've seen it. We will have this chart, okay? And it will ask, in which quadrant will you have the most single isolated colonies? In which quadrant will it be? 4, easy, you got it. Easy question on that. Okay, so no reason for anybody to miss that point. There will be a couple questions. I say a couple, but it will only be like a couple. That should be that. Okay. But anyway, so that'll be on there. Easy enough. There will be a microscope part and its function. Last time we had labeled the ocular lens. The ocular lens had that virtual. So the next time we'll have another part of the microscope, just know the parts and their general functions. But it'll be similar to that type of question. Like it'll label the part and the next question, what's the function of that part? Okay. So that'll be on there. Media basics. I'm less likely to ask a question about which media is solid or which media is liquid. At this point, we've advanced past that. And so most of our media questions are going to be about the biochemical test that we did. The stains, you do need to know the gram stain, the acid-fast stain, the endospore stain, and the capsule stain. So the 4 differential stains that we did will be back again. It will be the same slide that we saw last time, but it might be different questions. So if I asked about the primary stain last time, I might ask about the secondary or something like that. Gram, capsule, endospore, and acid-fast. There might be a simple stain question too. Don't remember. Okay, but it's fair game is what I'm trying to say. Biochemical test. So pretty much that's it. We will not have ELISA. We will not have any DNA questions. We will not have transformation. We will not have eukaryotes. No, we won't have any of that. So the stains, the microscopes, and then labs 12 through 18. Selective differential media and all that. So if you needed me to break down which labs specifically you needed to know, I would say safety, lab 1, 2, 3, 4, 5, 7. I won't be talking about the motility wet mat. Okay. I don't think you need to know that one for that one. And then you don't need to know Herbie Bauer. You don't need to know ELISA or on water or at eukaryotes or any of that stuff. So you don't need 8, 9, 10, or 11. Don't need to know those. 8, 9, 10, or 11 are out. Nothing about 8, 9, 10, or 11. Okay. Everything else though is fair game. We good? Okay. Okay. Also, when I set up the test this time, over here at this station over here where it's all in webinar, we have this station will be 1 question this time. Last time it was 2 questions. We had like sneak behind each other to get it. It will be 1 question. This will be 1 station. So you won't have to sneak behind the other person. There will just be 1 person here. So when you guys come in to start the test, one of you, and I don't care who, just find another empty spot to go to so that we can just have 1 person here. Yes, that'll make sense. And we'll go over it when we do the test too. But other than that, everything should be set up exactly the same. We'll rotate the same direction and everything. You'll have the arrows, you'll have the numbers. That part shouldn't be confusing. It's multiple choice and all of that. Same otherwise, just more content. Okay. All right, let's do it. Oh yeah, okay, talked about it. I could have just left it at that. Yes, we'll have 1 minute per station. And again, it'll just be between 1 and 3 questions at each station. If it's 3, I try to make them easier questions, right? We had the other one where it was like all the same choices, but a different question for them. So it'll be the same kind of format, between 1 to 3 questions at each station. Everything will be at our spots, and you'll have arrows and everything telling you where to go. 1 minute, keep your laptops in that little cubby, keep your laptops and your notes in that cubby. We did not have any issues with it last time, so it should be the same. What else? There's some other thing. And now my brain isn't wanting to remember it. Laptops. Oh, phones, right. Just a reminder, keep your phones on you, because if you need a duo push, I don't want you rifling through your bags or anything. Bags should go at the front or the back of the room, whatever you think front or back is, it doesn't matter. But front or back, try not to have them at our stations for the exam, because you don't know where you're going to be done. We don't want to interrupt people while they're at that station or whatever. So bags at front or back, keep your phones on you. They should be in a pocket, and they shouldn't come out unless you have, you know, a duo push. And I'll be there with you if you do. So again, we didn't have problems last time, so I think it should be fine. Okay. Oh, and I do want to make a reminder about the unknown report while I have everybody here. On the unknown report, you know, the 2nd and 3rd and 4th page where you have to like answer questions and type them in, not the chart that we have to fill out with our answers, right? It's not the result chart, but the other questions that we have to answer. The only part of that we should be answering with outside sources is the disease and uses. And the reason I'm bringing it up is you will lose points if you have more information than what we learned in the class, because I'm not stupid. I know where that comes from. We're copy pasting at that point. I don't want to see it. Okay. So I don't mean a lot of points. You might just lose 1 because I can't prove that you AI'd it, which is stated in the syllabus. You can't use it to answer your questions on your assignment. Okay. You can use it to help you figure it out, but your answers should be submitted in your own words. And so I want to be very clear on those, because I've had a few submissions, not saying it was anybody in particular, not saying it was any particular class, but I've had a few submissions that definitely are copy pasted from an AI source. It'll have like the symbols missing with like an X there and or a box there or something like that. Or it will talk about information that we didn't learn in class. So if you're putting information on there that we didn't talk about in our class that isn't in our lab notebook, You might be thinking, oh, well, this will look great because it'll look like I know more. Nuh-uh. Nope. It looks like you copy pasted is what it looks like. So, so don't do it because I will dock you a point for it, for each incident of it. Okay. Not 1 point overall, but each question that I think they did that on, you will lose a point. For example, if you're telling me about, lactose fermentation, let's say you chose to describe the PR fermentation test, and you're talking about how there is an export and import enzyme on the surface of the cells that allows the cells to bring in, called a permease, permease allows you to take in the lactose. We didn't talk about that in this class, so why are we putting that on the answer? That stuff, you get what I'm saying? Like if we didn't talk about it in the class, no, I don't want extra information. I want it only at the level that we learned it in our class, because that's showing me that you understand about the test, how I'm going to test you on Monday, right? And so do it in your own words. And I'm not being super strict. Like if you at your table, you decided to do PR glucose and somebody else at your table is also doing PR glucose, it's fine, just write it in your own. So you can share that info with your classmates, but write it in your own words too. So that's also another thing that could get you in trouble just for academic honesty type of stuff. So you used AI and you're welcome to use it to help you generate your answers. Just double check that you're not covering extra information. Just go back and be like, oh, we didn't talk about this. Take it out. Make sure your wording still makes sense. Make sure it's still relevant. Because then at least I know you're reviewing the information for what's relevant in the class. Because that's what the point of the assignment is. Yes. Did you have a question, Danielle?
Monday, that for the last question?
Yes, that's for the last question. Yes. Yeah. And for your survival questions, which is like the third question on the gram-positive and gram-negative test, be sure on that survival question that you are talking, well, on both the second and third question, be sure you're picking tests that are on the actual chart. I know I've said it before, but I've had multiple submissions where people are just like, the catalase test, the nitrate test, blah, blah, but none of those tests led you to your organism. So then I'm like, are you just telling me tests we did? Because I don't want to hear that. I want to know the tests that led you to the organism on our chart for this class. It's another way that I know that we're also just AIing that question. Because some of those tests like catalase or McConkey auger or EMB might be useful for identifying E. coli in laboratories in hospitals. But for our class, using our dichotomous key, which ones led you to your organism? So that will be your second question. The next question will be, explain one of those, how it relates to survival. So it should be coming from that list, and it should relate to survival. And if your organism was negative for everything, and that's how you got led to your organism, which is the case for like table 3, for example, they're gram-positive, had negatives, and that was pretty much it. Well, not making endospores tells you something about how it can't survive as well in the environment, and write about that. You know what I'm saying? So be sure you're following our dichotomous key. If you're like, hey, that dichotomous key was only given us to imply, that was one of the issues that I had with some students. We do know that this is on the assignment. Yes, this is in the lab unknown report assignment. There is a link to it on there. It's also in an announcement, a couple announcements, I think. So this is available to you. And if you didn't find it on your chart or you didn't have a picture of it or something to refer to, it is there on the assignment. Be sure it's only the test that we got there, because that's been probably the most common one that people are losing points on, is just giving me tests that weren't on that. Yes.
If I already submitted it, can I resubmit it? Because I
Yes.
didn't know.
So I think too, like I said to you guys that so I will say this right now is my cutoff for the feedback. If I gave you feedback prior to this very moment right here, or if you're learning from something I'm saying right here about something that you did wrong, or when you resubmitted it or something like that, okay? You are still welcome to make changes until the due date. But I won't be giving any more feedback after class today. Makes sense? I know I said 5 p.m. yesterday, but like I didn't get any feedback. So use the feedback that I've given you on your report or what I'm saying right now to help guide you with whatever changes you're making. And then pretty much from after this class moving forward, if I give you a grade, it's final. No more changes after this point, right? So on that, so I would try to grade them early, but if I grade it early and you got points off, that's not an invitation to resubmit it after this class, okay? We good? I hope so. because that ship will have sailed. And I'll make a comment about that too. I'll never mind, like, hey, you can't make any changes. This is your final grade in this FYI. So any comments that I make after this point are not open for you making adjustments. It's just me trying to get the grade out of the way. Okay. I like how you guys probably work the same way as I do. If there's something in your to-do list, you want to knock it out. It's the same for me. If I have things to grade on my print canvas, I want to knock them out too. So I don't like to wait to grade my stuff. I want to grade as much as possible. Plus, I know you guys appreciate having a grade sooner rather than later. I can't stand professors that don't grade their stuff within a week. Like, I don't understand it. I don't have respect for it. I don't know. What are you doing with your time? Because most of them are teaching online classes. So I'm like, anyways. Okay, yeah, that's my judgment talk for now for those professors, if that isn't clear. All right, so we're going to start with our culturing basics. This should be easy stuff. Bring it back all the way back to like lab 2. All right, first question. I want to start off easy. What is most commonly used to inoculate cultures? I don't even want to think about it. So most commonly we're using our loop. Of course, that's our inoculation loop. If we're doing a butt, we use the needle, right? So there's that. And many times I'll have media. So there are common questions on the test too. I'll just have a tube on there and ask, how do we inoculate this media? So we know we had some weird ones out there. The butts are always stabbed, right? The liquids we swish with the lube. But the slants, most of them we streak the slant. There was one that we stabbed the slant, and that was the TSIA. So be sure you know that one. Okay. We did streak and stab on that one. All right, so those are common questions too. So should be pretty straightforward stuff. All right, which quadrant of a street plate would you expect to find the most single isolated colony? I said it would be on the test. It's here in our review. Nobody will be missing this one. Yes, quadrant 4. It will be, I believe it's the first question, the first or second question. Okay. All right. So know about the generals about aseptic technique. Lab safety, be sure you wear your lab coat and your closed-toed shoes. And what do we do with broken glass? Sharps container. What do we do with contaminated disposables? In the biotrash. Yes. So stuff like that, right? Types of media, general concepts that the broths are liquid. The butts, the slants, and the plates are solid. What do we add to make things solid? Auger, right? Auger is what we add to make things solid. Different ways to inoculate the media. The stabs, the swishes, the streaks on the surface. It's pretty much only those 3 or a combination. Okay. Just there. Let's get into the more difficult stuff. Let's get into our stains. A lot of recall here. Any stain that we've done up to now is fair game. So let's see what we can say. What is the first step in the gram stain procedure? All right, first step is crystal violet. Crystal violet is going to get stuck in that peptidoglycan scaffolding. The next step is iodine. Iodine is the mordant, right? And it fixes the crystal violet into place. Then we do alcohol, and it's just ethyl alcohol, ethanol, to rinse out any crystal violet that wasn't fixed in place by the iodine. And then we counter stain. What's the counter stain for the gram stain? Safranin. Beautiful. So all the choices are up here. Just know what each one does. And if you leave out a step, right, what would happen? If it was supposed to be purple and we leave out the alcohol and it's purple now, is that accurate? Well, I mean, can you still rely on that purple being a positive? Or because the negative would look purple too if we left out the alcohol step. So it's unreliable if you leave out the alcohol step either way. But do know that they both would be purple if you leave out the alcohol step. We kind of learned that from the first one. Okay, what color do properly stained gram-negative bacteria stain? I say red because red is the color of what safranin itself looks like. It's a reddish stain. The cells, they look more pink, and I'm aware that they are more pink. And whatever works for you and how you remember it, but I won't put pink as a choice. Okay, so it will be something like purple or red or green or blue. Pink will only apply if I really feel like pink was the appropriate answer. So for example, if we talk about carbofuchsia, right, and we say something's supposed to be fuchsia, I usually won't put fuchsia as a choice because I feel like it gives away carbofuchsia too much. So I'll usually say reddish pink to describe that. And if you're ever confused and you're like, are you referring to fuchsia here? You can ask that question. That's a fair question. Are these cells fuchsia? And I will say yes or no. Okay. Or I might say, I think they're red. And then you'd be like, oh, okay. So that might reframe your thinking. So stuff like that is fair. That's fair. All righty. Next question. Sorry, I'm looking at this other screen. What is the name of this staining procedure? Our slide, we already know, it's not as clear as this. We've already seen our slide. It was one that most of us missed a lot on that lab midterm. And again, if you want to clarify what colors or what color or whatever's going on in the slide, you can always do that. So there in that image, we have red structures and greenish tealish structures. On ours, we got a little bit of a purple hue going on, but whenever I pointed out to you guys, oh, these are red and green, then you guys saw it. And it was like, oh, okay, yeah, those are red and green structures. It's just the colors were blending a little bit because it was an imperfect stain. So this is the endospore stain. The red structures are the vegetative parts of the cells, and the green structures are the endospores. If we were negative for an endospore stain, it would all be red. There shouldn't be any green structures. All right, which of the following bacteria will be acid-fast positive? A lot of similar questions for our review here, but we're just getting warmed up. This is an image of an acid-fast stain. It's actually one pretty similar to what we see in our class. So this would be Mycobacterium. We often get tripped up with this one. Remember, we're not going to be doing any tests for the mycoplasmas, but mycoplasmas, for reference, because we know it's going to be on the lecture final, mycoplasmas do not have a cell wall at all. So they would fall into the gram-negative category. Whereas acid-fast, we're looking for a thick wall with wax on the surface. So they're gram-positive. They're a subcategory of our gram-positives. And that wax is called mycolic acid. And it helps with survivability of that. It's not as strong of hardiness as an endospore is, but it's like in between endospore and like regular cell. So it makes them a little more hardy. So that's all going to always be Mycobacterium. Okay. So Mycobacterium pili, Mycobacterium smegmatis, Mycobacterium tuberculosis, Mycobacterium avium, all of those fall into that category. Mycobacterium. All righty. Next question. After the slide has been heat fixed, when do we use heat again in the acid-fast stain procedure? There are some questions on monthly lab final that if we are not well studied, if we don't know our information well, you would call it a trick question. I would call it, if you knew your stuff, you wouldn't think this was a trick question. And that's kind of what this is. We don't use heat again after we heat fix our slide with the acid-fast stain. We make the heat fix slide, and then with the acid-fast, we added the carbofuchsia for 5 minutes, and then we rinsed it off, and then we rinsed it with acid alcohol to remove the carbofuchsia that didn't stick. Why would the carbofuchsia stick? It's lipid soluble, and it binds to the mycolic acid wax. And so remember that one too, because we talked a lot about that one. It was on the lab midterm asking about the lipid soluble. It's the only lipid soluble stain that we have, carbofuchsia, and it reacts with the wax, mycolic acid. And so that's how that one works. And so we rinse it out with acid alcohol, and your acid fast, if you hold fast to carbofuchsia, the lipid soluble stain, with your waxes, whenever we rinse with that acid. You hold fast to the same, your acid fast. Okay. And the counter stain, what's the counter stain with the acid fast stain? You guys remember? Methylene blue. Methylene blue is the counter stain. So you're either blue or you're fuchsia. And again, on our lab final, it'll be a mixture. It was a mixture on our last one. Yes, we had blue cells, we had fuchsia cells. And then the question was something like, if this was a patient's sputum sample, what bacteria do they have? And it was Mycobacterium tuberculosis, yes, because Mycobacterium are acid fast. Commonly used in hospital settings to test for tuberculosis. All right, what color is a negative? Okay, what color is a negative acid fast stain? Let's see if you're paying attention. So our primary stain on the acid fast stain is carbonyl fuchsia. So positive cells would be fuchsia. Negative cells will be our counter stain. Look, exactly. Look, good job with that one. All right, what is the name of the primary stain used in capsule staining? So here's a really good image of what the capsule stain looks like. We got a big bold background. We got some reddish pink structures in the middle, and then white halos. A lot of people confuse this one for the Gram stain on the, I'm not naming names, okay, but I am saying we've got a big old bold background, and you can ask too. Is that all just that purple color or that bluish color? On ours, it was a little bluish of a color. Is that background or is that cells? You can ask that, that's fair, okay? So this was a background. We have white halos around reddish pink structures. So what are the reddish pink structures? If I were to ask that on the test, you would say they are cells that have capsules. Reddish pink structures in this, they're cells that have capsules. How do we know they have capsules? They've got the white halo around them. So remember, we're staining the background with the negative stain. This is Congo red. We then added the secondary stain, which was Meneville's stain. Meneville's stain is what made that red background turn purple. And it's also what helps stain our cells, that reddish pink color in that. Okay, does it make sense? We follow in with that. Any questions about the capsule stain? Okay. I definitely don't want the stains to be the things that trip us up since we've seen them, we've worked with them, we've been tested on them. So hopefully they should be old hat. Congo red is our primary stain. We have a secondary stain on that one, which was Meneville's stain. Remember also that the capsule stain was the one that we had to rinse in the Hoffman jars because we didn't heat fix it. The reason we didn't heat fix because it would cause the cells to shrink and it would create a false halo around them, false image of a capsule. So we don't heat fix with the capsule stain. All right, next question. In which staining procedure did we use steam? It was our mordant in that procedure. Right, so this was the endospore stain. This is the one where we had our slides up on the beakers, on the tripod, and we boiled the water while we added the drops of stain on top. What was the primary stain in that procedure? Malachite green, and it got everywhere. So that was malachite green. Secondary stain, safranin. Good job. All right, know our procedures. You don't need to know how many minutes or how many seconds. We kind of have an idea now of what I'm expecting for you guys for this. Know the general steps. What's the primary stain? What is the counter stain? What was the mordant? What was the decolorizer? What is the purpose? What is the primary stain doing in the staining procedure that's letting us get to the results? And how is it getting us a positive result? And what does that really mean for the structure of the bacteria? Having that reference as well. What's it tell us? If we stained with green structures in the endospore stain, we made endospores. What does that tell us about the organism? That it can create already dormant structures. It also tells us that if we made endospores, we must be a bacillus or a clostridium species. Like we had for table 1, their organisms, gram-positive was Lystinobacillus sphaericus, got bacillus in its name. We knew it would make endospores based on its name, and because it made endospores, we knew it had to be a bacillus or a clostridium. Same thing with table 2, they had Bacillus subtilis. They knew that if I told them that your organism was Bacillus subtilis, they would have known it made endospores. Make sense? Okay. All righty. Differential and selective media. I don't know details. I don't know what that means. You want to know what each is the media selective? Or is it differential? Or is it both? Or is it neither? Okay, so have that reference there. No, maybe that blood auger is differential, right? But it's not selective, it is enriched. So it's got growth factors to help fastidious bacteria grow. So let's take a step back from that, right? So what does fastidious mean? It means you need growth factors. You have things in your diet that you can't make for yourself. It must be supplied in your diet. Those things that we need supplied in the diet, we call them growth factors. And media that has the growth factors is called enriched. So the food that has the growth factors is enriched. The organism that needs the growth factors is fastidious. So remember which term goes where. That's often a common trip up on both of our last two things, okay? Because we know we're going to see that both in the lecture and in the lab. All right, so be aware of that. Is it selective? Is it differential? If it's differential, how is it differential? Is the media changing color or is the bacteria changing color? Because we remember, when you see something like this, this is EMB. So I should be able to hold up the plate by itself without seeing the growth on it. And you would tell me this is EMB because it's the darker colored one. But at the same time, I'm going to give you a plate. And you'll be able to pick it up and you'll be able to see, oh, it's got the metallic green on it. So what media is it? EMB. EMB was the only one that could have metallic green growth. So if I just put this plate, just to show you guys here, again, you'll be able to see it in the light and be able to see like there's metallic green on this. But if I just put the plate in front of you and say, what media is this? Just by looking at it, you should be able to tell me it's EMB. And then I might ask, on that media, is this organism positive or negative for using glucose? And you should be able to tell me that media doesn't test for glucose use. See how it comes off as a trick question. But if you know, what is this media, what sugar are we testing for using in this? Lactose. For both EMB and McConkey, we're testing for lactose use. And see how the actual organism changes color, not the media. So you'll have to know, the media is supposed to change or the organism growth is going to change. What color is it going to change? What do the colors mean? What is it signifying? So all of that are things that we need to know. I have some little kind of examples of that on this. If we're talking about mannitol salts agar, if we turned yellow, this was what it looked like uninoculated. This is what it looked like if it turned yellow. You also might have turned pink from alkaline. And we now have learned what was the pH indicator in mannitol salts agar? Phenol red. We've seen it a million times. It's always the same three colors, orangey red, pink or yellow. Pink is alkaline, yellow is acid, and it's almost always fermentation. So what did we ferment in mannitol salts agar to turn it yellow? What sugar was it? It was mannitol. It was mannitol. So that one's in the name. Okay. So yes. So those are based on phenol red, just like our PR fermentation tubes and the urease and the TSIA were. You good? Okay, so we have some examples of that, just showing you guys so you can see what I'm talking about with mannitol salts agar. It's this one here. This is an example of one semester, we did not get the media made because I think I told you guys about this, that our DI water for the whole building was at pH 3. And so none of our media made properly. And so I had to just get pictures for it. So this is left over from that semester. So we'll kind of refer to this while we're doing our review today, since the media doesn't keep, and I don't want to inoculate it until exam day. So we have it then. You may or may not, depending on how my bacteria behave when I inoculate them, which we learned about this too. You may or may not have a picture or a plate, okay? But they should be clear, whichever one I use. The reason I will choose a picture is because my plate was iffy. Okay. All right, here we go. Let's do it. First question, what type of media is pictured? Okay. I didn't even look at it, so I have nothing to say. Let me see. Okay, yes. So just like the one that I showed just a second ago, if we have metallic green, this must be EMB. Right? I mean, I'm going to be completely honest. If you have EMB on your test, it'll be the one with the metallic green because I feel like that's really what distinguishes it as different from McConkey or the other ones. Because EMB is the only one that can have metallic green. Okay? Can EMB have a pinkish color of the growth? If your organism just used lactose normally, then the growth will be pink. But if you aggressively use lactose, then it'll be metallic green. Okay? All right, good. Easy one to start with. Let's see if we get that one. What is the selective agent in this media pictured? And EMB, what's the selective agent? So we have differential and selective for this media. What's the selective agent? So selective agent is something we add to the media to prevent some things from growing and allowing others to grow. In EMB, it's eosin methylene blue. It's what EMB stands for, okay? That's our selective agent there. What type of media has biosalt as the selective agent? Anybody remember? McConkey, yes. So McConkey has biosalt for the selective agent. They're similar. They both test for lactose use, but EMB has eosin methylene blue. And if you're an aggressive lactose user, you'll turn metallic green. McConkey has biosalts, and if you use lactose, it'll only get pink. It will never progress to the metallic green on McConkey. Okay, so they're different selective agents. They both allow for what type of bacteria to grow? Gram negatives. So gram negatives grow on EMB. Select to prefer growth of gram negatives on EMB and McConkey, typically. Okay. All right. Which media is this? It's pictured. So we've got a reddish pinkish background, and I'll say, let's call this pink growth on it. Reddish pinkish media with pink growth. This is a little bit of a tricky one. On the test, I'll try to keep it a little bit clear. This is McConkey agar. Okay, this is McConkey. So the difference between the actual colors of the media of McConkey versus EMB, EMB media is that darker color. McConkey is like almost looks like phenol red media, like a reddish orangey color, like pinkish reddish orange color. Okay. FYI, I am less likely to just put something like McConkey up in front of you and ask you what the media is like this because it looks so similar to other media. So I'm more likely to say, this is McConkey agar, and then did this organism, metabolize lactose? Is this organism likely gram positive or gram negative? Something about it and give you context. More likely then. But if for some reason you're still confused about it, you can ask me questions. Now, can you ask me, is this McConkey agar? But if you give me some context of what you're thinking and what line we're along, then I can help you out with helping decide, okay? It shouldn't be that complicated. I tried to pick things that were obvious, okay? All right, next question. Select all that apply. Is the media that's pictured selective or differential? So this is McConkey agar. Is McConkey selective or differential? Or is it enriched? Or is it fastidious? Or is it a multiple? So it is both selective and differential. What's the selective agent in McConkey agar? Biosalt. We thought just a little bit ago. So biosalt is the selective agent. How is it differential? You can grow on it or not. If you grow on it, if you turned pink, you use lactose. If you just grew on it and didn't change colors, you did not use lactose. That's the differential side of it. Selective and differential. It is not enriched. Our only enriched media that you have to know is blood agar. It was the only one that was enriched. Okay. And remember, the organism will be described as fastidious, not the media. We're doing great. What media is pictured here? Again, just based on this picture, you should be able to know what it is. That's a pretty clear, pretty obvious picture. There's not much questioning about what's going on this one. This is mannitol salts agar. How do we know this is mannitol salts? Because the organism turned the media yellow. And so what happened here? The organism in the yellow area on this media, it fermented mannose and it made acid as a product and it turned the phenol red, that yellow color. We see this organism here, how it turned it kind of pinker. Well, that organism broke down the nitrogen source and made ammonia, as per we always talk about all the time in this class. It used a nitrogen source. It grew, clearly. Did it use the sugar in there? Probably, aerobically. Didn't ferment it, didn't make acid. And the alkaline product from using the nitrogen is what turned it that pinkish color. Are we tracking? Is it coming together? Are we seeing the big picture of these results? I hope we are. We're getting there. All right. Beautiful. What is the pH indicator used in mannitol salt agar? We're dealing with red and pink and yellow. It's always the same thing. All right, so this is phenol red, phenol red. So know which media is selective or differential or enriched or both or some combination, all right? Those are common questions. Know what the selective agents are and what they're selected for and how they work, right? General concepts there. I don't need you to explain the chemistry of how lysine methylene blue prevents gram-positives. I don't need the chemistry of it, but just know that it does, because that's all we talk about in our class, okay? We talk about the salt in Staphylococcus agar. You should be able to tell me the salt favors gram-positive growth because gram-positives have a thick wall to help protect against osmotic pressure. You don't have to use the word osmotic pressure. I have an idea of what that means. Salt causing water to get leached out of the cell, causing it to change shape. What's that process called? If we put something in a hypertonic solution, high salt, a paclitaxel salt, it draws the water out of the cell, causes it to shrink. Does anybody remember the term for that process? Plasmolysis. Plasmolysis. That's what it's called, yes. That isn't going to be on your lab exam, but it will be on your lecture exam, probably, maybe. Okay. Possibly. I don't remember all the questions, okay, but you need to know it for the lecture thing. All right. What does each color change mean? If we see pink growth on McConkey or on EMB, we use the lactose. If we see metallic green on EMB, we aggressively use the lactose. If it didn't change color, we didn't use the lactose on McConkey or EMB. We're talking about mannitol salts agar, then we need to have reference for mannitol salts agar. It started off with a reddish orange color. If it turned yellow, we made acid from fermenting mannitol. If it turned pink, we didn't ferment mannitol. That was the most important aspect of that one. And that sort of stuff. In blood agar, we need to know the three types of hemolysis. If we just grow on the blood agar and there's no change, that's gamma hemolysis. It's still hemolysis. Don't ask me why, okay? But it's called gamma hemolysis if it doesn't change. If we have slight breakdown, but not complete breakdown, and it's a little bit greenish, that's alpha hemolysis. If we have complete breakdown, we have clearing on the blood agar, and that is complete breakdown of the red blood cells, total hemolysis of blood cells, that is beta hemolysis. We tracking? You have heard these terms probably in some of the presentations or in some of them that are coming up. Some people will talk about their organisms, and if we read in our chapters and learn our stuff for chapters 19 through 24, some of our organisms are called like alpha hemolytic or beta hemolytic. That's based on the results on blood agar and what type of hemolysis we see. So alpha hemolytic strep or beta hemolytic strep or alpha hemolytic staph, whatever. So those are different aspects that we often use to compare and contrast some of these pathogens. Any other questions about the differential and selective media? Anybody want me to cover anything more as we moving on? Anybody online want me to cover more? You can always email me. I want to say it's not, speak up now or forever hold your peace. You can email me between now and Monday. All right, biochemical tests. Now we're moving on to our PR fermentation, our MRVP, our litmus milk, urea, all of those. Okay? Which test is this? Believe it or not, this is a common question on my exam. Just to have some tubes and ask you what test is it. So this, I will say, is this one. This is slant. Okay, that's not clear from the picture, it's a slant. So this is what our actual tubes look like. Right? So these are the literal tubes that you'll see on the exam. These are the actual tubes that we'll see on the exam. Now, in my hand, so we have from your perspective, this is your left and this is your right. Which tube is positive result? This one. Good. So if I were to say which one was uninoculated? All right. What happened to the pH in this tube? Went up, alkaline, right? And then what test is this? Citrate or citrate. So how do we know this is citrate and not malonate? So I didn't put malonate on here, but you better bet you're going to need to know the difference on my exam. Malonate's a broth, exactly. What are we testing for? What did this organism do that this organism didn't? This organism was able to use citrate as a carbon source, right? So this organism in the blue tube can use citrate as a carbon source. This means it used citrate for carbon and it broke down the nitrogen source. Ammonia was released. Ammonia is alkaline. What is the pH indicator in this tube? Bromothymol blue. Bromothymol. And don't just rely on the B one, because I might have words that look like the word. Know the word, okay? If I put bromophenol blue, don't fall for that. Hint, hint. Yeah, like phenol red, exactly. It's phenol red, right? It's bromothymol blue. And what was our third pH indicator that we used in the class? Litmus, easy one, should be easy. Yeah, there's only one test. It looks like milk. Like we all kind of knew that one, okay? As difficult as we thought litmus milk would be, there's a lot of stuff that points at it being milk. All right, we good on the citrate test? There might be one or more questions about this. Who knows? I don't have this memorized. Yes, which of those tubes was positive? If this was A and this was B. A, B. Sorry, it switched it up, okay? Yes, yes. So the blue one. Good job, guys. All right, what's happening to the pH in tube B? We covered that, but hey, it's not bad to just keep saying the same thing over and over again.
That one was the red slant.
Yes, that had all the different colors that were happening in it. Yes. The pH went up, right? Alkaline from ammonia, from using the citrate allowed us to use the nitrogen source and then make ammonia. Okay. All right, so how do we inoculate the citrate plant? Normal streak on the surface. This is just a normal streak on the surface of the slant. How do we read it? If it turned blue, we used citrate. If it stayed green, then it didn't use citrate. What colors are produced? Blue. What does an uninoculated tube look like? Green, okay? What do the colors mean? Blue means alkaline. It means that we use citrate, which let us use the nitrogen, which let us make ammonia, which is alkaline, causing our pH indicator to shift colors. What was the pH indicator? Bromothymol blue. We good? Any questions about citrate? Okay. We're going to try to break them all down as much as we can. Which test is pictured here? Uninoculated one. It's got a Durham tube in it. Here's some examples that we might see on the test. Liquid, Durham tube. But we should know this one just by looking at that one too. These are some possible results. This test, I usually always ask about it in reference with an uninoculated and an inoculated tube. So you usually won't just see the one tube for this one. So that was an image of the uninoculated version. This is the phenol red carbohydrate fermentation tube. You do not need to memorize that the blue one is the glucose and the red. I don't need that. I will give you reference on which one is which if I need you to have reference, okay? Otherwise, it should be enough to know this is the phenol red carb fermentation tube. Then I might ask, did we ferment glucose in this tube? And it won't be a trick. You don't need to know that this is not the glucose. Okay, that won't be meant. If I say this is the glucose PR fermentation tube, you know, did this organism ferment glucose? Did this organism ferment glucose?
Yes.
Yeah. Yeah, we've got acid produced here. And if I ask in this tube, and again, you would need to look at the tube. You will have the tubes in front of you, and you may pick them up. Please do not spill any of the tubes, okay? A little bit of preface here. If one spill happens in any of my sections, if a spill happens after that, you will know because I'll be cranky, number one. Number two, people go and lose points, okay? So I will usually give leeway for one spill, but then after that, listen. The only reason I'm bringing this up is I had a semester where people kept spilling tubes. I had 3 tubes get spilled in one section in one class. I cannot tell you the level of frustration that I was at that point with it because, you know, we already had a talk after the first spill. And I'm like, how is this still happening? Like, are we serious right now? So please just be mindful. We know these are not sealed. These are not capped. And there are tests where you will need to tip them. But if you need to tip them so far that it's going to spill out, you should get your answer. Okay. You should have your answer without having to spill it. For example, why would we need to tip the tube? What 2 tests might we be looking for solid or liquid on? Gelatin, obviously, gelatin test, right? Is it liquid or not? The other one will be the litmus, right? Because some of our litmuses might go, yes. You see how this one is chonky? So you tip this a little bit, you can see that this one is solidifying. Yeah. So that helps with that stuff, looking at that. So this one's like almost gone fully solid with that white stuff in there. We just got this curds and whey now. Yeah.
Ew.
I know. Curds and whey now. All right. So anyways, be careful when tipping your tubes. Don't spill them because some of them are real stinky too. And that ends up being stinky. And we know you're just spreading that bacteria at every station that you're moving to. Let's just not do it. All right. So phenol red carbohydrate fermentation test. Let's get into our questions. What is the name of the upside down tube? Usually by this part of the course, this is an easy question. It will be on the lab final. Okay, that upside down tube is called a Durham tube. What's the point of the Durham tube? To collect gas from fermentation typically. So gas from fermentation, what's the point of the Durham tube in the nitrate reduction test? Collect nitrogen. Still same similar kind of idea. They're both involved in fermentation or in respiration. Yeah. Remember that fermentation is a type of respiration. Okay. Right. So that's what that tube is. Durham tube. All right, next question. Select all that apply. What are the 2 products of fermentation detected by this assay? So again, both products are shown in this tube. If I were to ask you what products were made in this tube. All right, so they're both visible in this tube. We can detect acid, which is a product of fermentation, and we can detect gas, which you see as gas collected in the top of the Durham tube in there. Any questions about that? Now, if we were to see a pink tube like this one is pink, and I'll give you an uninoculated one so you'd be able to compare it and tell me that it was pink if you saw them next to each other. Is the pink a product of fermentation? No, aerobic respiration usually. Okay. And why is that? We break down peptone, make ammonia, alkaline shift, causing this to turn pink. We good? Not fermentation though. All righty, which test is pictured here? I don't have it up here, so. Oh, I do have that. But I often show the test in a triple. So well, I'll either show it in triple or double depending on what questions I'm asking. So this is a common one that I'll see on the test. You will have at least 3 questions about them. 2 stations, at least 3 questions. I think there's 4, maybe 5 questions about this one, SIM. How do we know it's SIM? Because of this red band at the top here. Why did this turn red? What reagent did we add here to get that red band? Kovacs reagent. What were we testing for? Indole, which is a product of breaking down tryptophan. So we following, so we're getting into SIM. It's coming. Okay. I often will give this with uninoculated tube and then the inoculated. And then you would tell me like what products are in it or whatever. Okay. If I ask what reagent was added to the tube to get that result, you should be able to tell me it was Kovacs. Okay. What does the I stand for in SIM? That is one that I usually have to make fresh tubes for, which is why I don't have good examples for this one. So, all right. So I stands for indole, which is a product of breaking down tryptophan. Okay. So if I ask a question on the test and I say, which of these 3 tubes broke down tryptophan? Which organism broke down tryptophan? We'd say the organism in the middle tube, right? Have that perspective. All right, how is indole detected? What do we add to it? That's what the question's really asking. How do we detect indole? We add Kovac. In what test do we add zinc? The second step of the nitrate reduction, right? What test do we add peroxide for? Catalase. Good. What test do we add FeCl3? I know. Phenylalanine deaminase, right? Okay. So that's where we use those other reagents for. Okay, so Kovacs is for the SIM media to test for indole. Indole is the byproduct of tryptophan. The repetition. Okay. How do you detect H2S in SIM? Which of these two have H2S? Right? So we'll observe a black precipitate. Which of these tubes has H2S? This one here, right? This one with the black precipitate in it. Obvious. It will be that obvious on the test. I won't have like a little speck of black. It'll be one that looks like something like this. Okay. So this is H2S. What is H2S? It's a product of sulfur reduction. So if we reduced sulfur, we make H2S as a product. How is the black precipitate present in this? The H2S reacted with an iron compound in the media. This is true for SIM and for TSIA. The I in TSIA stands for iron. And it's iron that's in the media. It reacts with the H2S and makes the sulfur product. We don't need to know more about the chemistry than that, okay? I promise. I mean, Dr. Tim would probably be sad, but I am not, I don't care enough, okay? Not at the level that you guys need to know it. You're not going to be microbiologists. We don't need it, okay? All right. How is motility detected in the SIM test? Again, I'll often give you 3 tubes that you can compare to each other. I'll have an uninoculated and 2 inoculated tubes, and you'll tell me which of the inoculated tubes is motile or non-motile, depending on what the question you're asking. Okay. And they'll all be labeled A, B, or C, and you'll just tell me tube B is motile or non-motile, or whatever the question is. So it's growth away from the stab line. And typically, mine aren't too bad on what I have left up here, but typically, like if you see the stab line and it is clear around the stab line, non-motile. If it is foggy, or if you can't see the stab line, but you know that it grew because you can see bacteria are there, fogginess, and it should be on the test, it should be foggy, okay? The one that I walked around in class the other day, it should be similar to the one that we saw in class. Okay, so clear stab line versus fogginess in the media, okay? Fogginess is motile, clear stab line is non-motile. We have questions about that one? Okay, I know it seems obvious, but I remember on the test, if you're looking at it and you're like, I think this tube is cloudier than this one, I can verify that for you, okay? I can't tell you that the cloudiness means motility, but I can tell you it's cloudy. All right. So how do we inoculate them? Stab the butt with the needle, and that's it. Just one stab on the butt. How do we interpret the results and the change? We went over them pretty good. For sulfur reduction, H2S is detected when it binds with iron and makes a black precipitate. For indole is detected when tryptophan is broken down and makes indole. We add Kovacs reagent and interacts with the indole to make a red product. What does the negative look like for indole? It's just going to say that golden layer on top. What reagents do we add? Just indole just Kovacs to detect indole. We good on Sim? Any questions on it? I'm checking chat too, so if you guys end up talking in chat, I'm going to keep my eye on it. Which test is pictured here? This is an uninoculated tube. Yes. And I'm sure when we started all of this, we were like starting to get a little confused about these different things, but now litmus really stands out. It's a lot different than the other tests. So this is the litmus milk, and I'm glad we agree. You guys already know what's coming. We got our line up here. We got our line up here. We got our line up here, right? Okay, on the test, I won't have the whole lineup. On the test, I'll have an uninoculated tube and one of these, okay? And I usually only have 2 questions about litmus on the test. Not gonna be a ton of litmus questions, okay? There might be 3, okay? I don't know for sure. 2 or 3 questions about litmus. All right, in litmus milk, what color is associated with an increase in pH? That's the uninoculated. We know it starts out as lavender. What do we see if we increase the pH? Just increasing the pH first. Nothing else weird happening. Increase in pH. Increase in pH means alkaline. This is our right. This is our lineup. Okay. This is our lineup. Darken the camera, but you guys can see it, okay? If this is our starting point, that's our lavender starting point. Get this chair out of the way, okay? This side is acid side. This side is alkaline side. Okay. What's happening on the acid side is we're fermenting lactose. What's happening on the alkaline side, what's always happening with alkaline? Breaking down nitrogen sources, which in this case is proteins and milk, okay? So on the acid side of things, pH is going down and even further down. Okay, so this is like our pH scale, if you will. Acid side, alkaline side. Acid is pink. Acid is pink. And then even more acid, we've now reduced litmus. And so this tube's been sitting here long enough, there is no sign that litmus was ever in here. It's gone completely white, and we have coagulated the milk as well. We've curdled it. And so it is now solid in there. And we have liquid and solid portion. And the liquid portion, we call it whey. And the solid portion, we call curds. And we make cheese. I mean, that's what's happening in here. Yes.
I have a doctor.
Yeah. Go. Got it. Point me. You gotta go. All right. So that's it. And we're recording this too, so no worries. And I'll post it later so you can see if you need it. So yes. So acid from fermenting lactose. If we really commit to that, we can actually reduce litmus. This is reduction. Okay, so if I ask what's happening in this tube, you would say reduction. You would also say coagulation. Can I see any gas produced in here? I can't see any gas produced. There's a little bit of bubbles in it. Maybe. I'm not going to ask questions about the gas, okay? It's just hard to see it. But anyways, that's what's happening in those tubes, okay? For the alkaline side, the alkaline is where the pH is going up, right? So in these 2 tubes here, the blue tube is an alkaline shift. This is from breaking down, usually casein. So we have several proteins in milk, casein, whey, and all sorts of stuff. So here, if we're breaking down casein to make ammonia, then we get an alkaline shift. If we're breaking down all of the proteins in milk, then we get digestion. And that's what that looks like. So those are the different things in litmus. I'm sure we're going to have more questions about it on here. So we're going to go through it and see what we can say about it. Before we get to the questions, do you have any questions about litmus and the pHs and everything like that? So I put this tube in front of you, this pink one, okay? And I asked you what's happening in this tube. You would say acid production from fermenting the lactose. Okay? If I asked you what's happening in that tube, you would say coagulation and reduction. If I ask you what's happening in this tube, alkaline from breaking down the proteins, making ammonia. If I ask you this tube, you would say digestion. Make sense? Okay. So on the test, I will have 2 tubes, an uninoculated one, and one of these other 4. And then you'll tell me what's happening in the tube typically. I might ask you what the pH indicator is. What's the pH indicator? Litmus. Okay. All right. Let's see. Yes. So Deontay is asking a question. He says on the review, like those study slides that I have for you guys, one of the study sheets has a chart and it says SIM is a slant that's inoculated with a loop on the surface. It is not. SIM is a butt, and that's a mistake in that study sheet. If you guys see any other mistakes in the study sheet, let me know. But I feel like SIM is one that we are all pretty familiar with, it being a butt that we stab. Okay, so it's a butt and we stab it with the needle. Yeah. Remember, because SIM is the sulfur, the indole, the motility, we had to stab it in order to get most of those results. So good question. I'm glad you asked that. All right, in litmus milk, white color, what does it mean? White. And I don't mean a little bit. It's white. Look at the whole thing. Or at least like 9%. So that is reduction. Reduction. Do we also have coagulation in it? Yes, but the white color is the reduction. The coagulation is the chunkiness. So it's just a terminology issue. That's a semantic thing. Okay. And you won't have coagulation unless you have white. Okay. So they come together, but they do mean different things. Interpret the colors, what's happening with the pH, what is actually happening with digestion. We're breaking down the proteins, all of them in the milk. How it's gas detected. We would see it as cracks or bubbles in there. I'm not going to test you on that. It's not on the test. Okay. The sugar in milk. What is the sugar in milk? Lactose. Lactose. All right, let's move on. Which test is pictured here? Let's see if we can find it. Oh, I don't have an example yet. This is as good as it is. Definitely mostly looking at this top line here. Thank you, Darren. All right, so that's the starch hydrolysis test. We know because that first place, what it would look like initially, then we add a reagent to it and it causes the starch in the media to turn this dark purplish brown color. What reagent do we add? Iodine, yes. Iodine reacts with the starch. So if you create a halo around the growth, so here we see the growth of the bacteria is in the middle, and then we have this glow around it. That's where the bacteria has broken down the starch and so caused it to have that kind of glow around it. And this is just a different test going on the bottom. We don't look my copy and pasted it, okay? So the different test on the bottom is not what we're talking about. We're talking about the test on the top. So we added iodine. This organism, did it break down starch? Yes, because it's got that glowy halo around it. What's the enzyme that bacteria make that breaks down starch? You guys remember it? Amylase. Good job. Amylase. Okay, easy enough. Probably going to ask us what the reagent is. Okay, which organism is positive in the picture? Sure, we're reading our choices here. right, so that is gonna be the one on the left here. We've got that halo around it. And on the test, I'll make sure it's that obvious if it's on, okay? Some of the questions on the test, I might not even give you the plate, but I might just ask you, what would it look like? And then you have to select of what it would look like. That often happens if I can't get it to grow properly. And pictures and that stuff. What is added to detect starch hydrolysis? Easy, easy peasy. What reagent? Alrighty, so this is going to be iodine. Good job, guys. We're doing good. All right, which test is pictured here? Okay, so this is a tube like this. It's got a Durham tube in it. Liquid, I would say it's plain media color liquid. The Durham tube. Plain media color tube. It has a Durham tube in it. This is the nitrate reduction test. We just did it on Monday. Still feels like the same day to me. That's okay. All right, what do reagent A and B detect in the nitrate reduction test? If we add A and B, what are they detecting? I do have it up on the board, but let's see if you can remember. This is one that I left on the board just because we need a lot of practice with it. We add A and B. All right, A and B detect nitrite. What do we add to detect nitrate? Zinc. Excellent. And that's in the second step. Yes. So A and B first detect nitrite. If you don't turn red, so remember red means nitrite and you're done. Did you reduce nitrate if you make nitrite? Yes, and you'd be done. However, if we add A and B, no color change. We don't know yet if you reduced nitrate, so we have to add zinc to test for nitrate. If you turn red with zinc, did you reduce nitrate? No, excellent, because nitrate is still there. At that point, if we add zinc and you still didn't change, then you reduced nitrate. It's not nitrite, it's some other thing. Not nitrite, it's not nitrate, it's something else now. Who knows what, because we're not going to test it and we're not going to worry about it. Okay. Nitric acid, you know, that sort of stuff. Okay. What does zinc detect? We asked it, let's see if you remember. Zinc comes late because we're testing for nitrate. I remember it's that stupid, but it works, okay? Okay, yes, zinc tests for nitrate. And if you can remember that, and you know, only thing that we're testing for is nitrite or nitrate. And if zinc comes late in the alphabet and it comes late in the test, it tests for nitrate because it's wrong. And you'll remember that A and B test for nitrite. Okay? Lean into it. It's silly, but it works. All right. Know what is detected at each step. A and B detect nitrite. Zinc detects nitrate. Red is not always positive for the big test, right? The overall asked question is, did we reduce nitrate? In the first step, red means yes. In the second step, red means no. So it just depends on what we're doing. Don't forget gas. You could make N2 gas and collect it in the Durham tube. If you had gas collected in the Durham tube, I don't need to add A and B. I don't need to add zinc. I would know immediately that we reduced nitrate to N2 gas. None of our tubes did that. You won't see that on the test. No, you won't see like physically on the test, but know the idea of it, right? Okay, any questions about nitrate before we move on? Because that's all the review we're getting on it. We just did it on Monday, so it's just a refresh in our mind. All right, which test is pictured here? Let's see if I have it. Oh, I don't need it. That's a good picture of it. Which test is this? A whole bunch of different possibilities with the results. Good picture. I have several examples of it. Yes. All righty. This is the TSIA. And it looks like this. Nope, I don't have it up here. Here. It looks like this when it's uninoculated. It's got this orangey color to it. And I will, on the TSIA, almost always give you uninoculated and inoculated. So that way you can see if it's pinker or not. Because that's usually where people get lost. Will it pinker? I don't know. So you can compare it. Okay. So it'll be there for your comparison purposes. Cool. A lot of different options here. We're going to ask about the different ones, obviously, in the next 10 questions. Select all that apply. Which tube or tubes show evidence of H2S production? Yeah, so H2S production, we're looking for black precipitate here. So remember that H2S interacts with the iron. Remember, TSIA stands for triple sugar iron assay. And so if H2S interacts with iron, it makes a black precipitate. What is H2S? A product of sulfur reduction. So the black precipitate is indicating sulfur reduction. So here we have tubes 3 and 5 at that. These really smell bad. It's just getting worse, I feel like, the longer I sit here. Which tube shows evidence of gas production other than H2S tubes? Not 3 or 5. Which other tubes show gas production? Right, so if we look at this, we see, if we were to pick up these tubes, you'd see a little bit of gas in tube 3 and 5 here. There's a little pocket of air in there, but it's hard to see on the image, which is why I left it out of this question. But obviously tube 4 and 4A, we're making a lot of gas there. That's causing the media to lift it up out of the tube. So that's tubes 4 and 4A on that when we're making gas, okay? Let me see. I don't remember what the next question is. So how is it inoculated? How do we inoculate TSIA? Streak and stab. This is the only one that we do 2 types of inoculations on. It's the only weird one. So we streak it and we stab it with the needle. How do we read the results? With our uninoculated tube, looking like this. What's our pH indicator in this, first of all? Phenol red. Once again, phenol red. So again, what would yellow mean? Acid. What's the acid from? Fermentation. Fermenting what? If it's acid in the butt, fermenting glucose. If it's acid in the slant, fermenting sucrose or lactose. So remember that, slant, sucrose, lactose. And that should give it away, slant, SL, sucrose, and lactose. But is glucose. I don't have a good one for you yet. Sure, gas, because we're often do the gas. Why not? So yeah, I don't know, whatever works for you, but the butt is going to be glucose, okay? The slant is sucrose and lactose. And it depends on what the color is and where it's happening. You can also see alkaline. If you see alkaline, it's usually from aerobic fermentation. The aerobe, whatever it is using the nitrogen source, which is peptone, just like in our glucose fermentation tubes. Peptone was in these. And so if we break down the peptone, the nitrogen source that's in the TSIA slant, we will make ammonia. And now increase the pH, increase in pH, alkaline, it's going to turn pink. All right, so that's the possibles for the pH shift. We also can have color change from black precipitate, and that's from sulfur reduction. If sulfur is reduced, we make H2S. H2S binds with the iron, and that causes the black precipitate. Any questions about any of that? Do we add any reagents to this? No reagent. Okay. Just checking I'm not missing anything on chat. Which test is pictured here? These are the tubes that you'll see on the test. These are the actual tubes that could appear on the test. This is the urease. If this is on your right, left, if this is on your left and this is on your right, which tube is positive? Right? Okay. What happened to the pH in this tube? It went up. Okay. So what product did we make that made it go up? What chemical was made in this that made the shift go up? You guys remember? Ammonia. Okay. If our pH is going up, it's ammonia. In this case, if your organism, so in the pink tube, show you guys, in the pink tube, pH went up, we agreed on that because phenol red. That's our pH indicator, yet again, right? It's a watered down version of phenol red, but it is phenol red, okay? pH went up in here from making ammonia, and I'm telling you that. But what happens is your organism makes the enzyme urease. The urease breaks down urea into carbon dioxide and ammonia. And so that's why the ammonia is made here. That's the pH issue there. Are we tracking with that one? Okay. So it's probably going to ask us what the pH indicator is. But we already know now. Oh, no, it's not. I see by a different thing now. All right, what's the next test? We got some bubbles fizzing on a slant. Now we'll see if you guys were paying attention, if you know the difference between these tests. We got bubbles on a slant, y'all. That is catalase. I don't have a good way to help you remember this one. To me, it's easy because I remember cytochrome C oxidase. I just lean into cytochrome C oxidase. I remember that whole blob of a word, and that has oxidase in it. And so that was the purple swab test. Catalase catalyzes converting hydrogen peroxide into water and oxygen, and that's where the bubbles come from. Are we good with that? It's probably going to ask us what reagent we added to get the bubbles. No, it's not. We're on to a different test. All right. What test is this one? If our last test was the catalase test, this is the oxidase test. You don't need to know the chemical name of the reagent for this one. For the catalase test, the reagent was hydrogen peroxide. This was just oxidase reagent. Okay. Purple is positive, so we see that one on that. I think that is. Yes. So other tests that you'll need to, that's it for those ones. Other tests that you would need to be familiar with, malonate. Malonate was like citrate, but broth. These are the tubes that you would see for malonate. It's a broth version. Which tube, left or right, is positive? Yes. So this, again, so to show you guys, these are the tubes that will be on the test if you see them. The blue. Now, what happened to the pH in the blue tube? It went up because alkaline. What's the pH indicator in malonate? Bromothymol blue. Same as citrate. It's the same thing. It's just a different carbon source in a different form. We're not slant or broth. So if I give you on the test these two, and I say which one, left or right, is the citrate test. Yes. And the malonate is this one. To know the difference, be able to know that, right? I often just give the tubes and ask you what test for these ones. I might also ask what's happening to the pH or what's the pH indicator or something like that. But having that reference on that it's citrate or malonate, you'll need to know it by sight. Okay. That's the malonate test. We got that one. Trying to keep them separated here so I can find them later. MRVP. The MRVP test looked like this uninoculated. And then we had to separate it into two tubes. And then we added methyl red to test for heterolactic fermentation. What color did you turn for the methyl red test if you had heterolactic fermentation? Red. Those ones, red is positive. Okay, same for MR, same for VP. If we add methyl red and it turns red, you're positive. If we add methyl red and it turns orange, you're negative for heterolactic fermentation. For VP, what does VP stand for? Voges Proskauer. And it's testing for mixed acid fermentation. We added 1 naphthol and potassium hydroxide. If it turned, we had to let it sit for 30 minutes. You guys remember that one? If it got red at the top, then it was positive for mixed acid fermentation. If it didn't turn red at the top, it was negative for mixed acid fermentation. That's the extent of what you need to know for MR and VP. They are separate tests. They're testing for different aspects of fermentation. We good on MR and VP? Okay. Gelatin. Was a butt that we had to stab, okay? And don't worry, it won't like melt in the room accidentally. If it's liquid, it was meant to be liquid. If it's solid, if it was solid, it would be solid, okay? Don't worry about that. We may have tricked it, we may have rigged it, so that should be obvious, okay? So for example, if I were to say, this is your left tube, and this is your right tube, yes, okay? Which tube was positive for gelatinase? So this is liquid.
Because it breaks it down.
Yes, because it broke it down. Exactly. So it was solid, and you'll need to know that. So if I just give you the two tubes, which one's positive for gelatinase? Gelatinase breaks down the gelatin. That made it solid. Remember, gelatin is a solidifying agent. Make sense? Are we tracking that? And it will be this different. You can tell that that's liquid. And so you should only have to like shake it a little bit and be like, okay, that's liquid. It's not tricking you. It really is. Okay. So you should know it. That pretty good? That pretty clear for you guys? Okay. Put this back so I don't lose it. Okay. And phenylalanine deaminase. It was a slant. I don't have one to show you guys because I have to make that one fresh for the test. It doesn't last. So that was one that was a regular slant looking slant. And we had to add ferric chloride, which is a yellow oily liquid, to the slant. If it stayed yellow, you did not deaminate phenylalanine. If the slant turned green, you deaminated phenylalanine. What does deaminate mean? Remove an NH2 group. Remove an amine group. Okay. So what color would it be if it was positive for phenylalanine deaminase? Green. Green slant. The slant will be green. Are we good with that one? Okay. So it should be a pretty obvious one. And on the test, it will be, like you'll see that it is definitely green. Okay. And then if I have a tube that's sitting there with a green slant with some liquid at the bottom of the slant, and I ask you what reagent did I add to it, it will be FeCl3. I'm not going to try to make it a trick, okay? It'll be FeCl3. I'm not going to put some different variation on it, but you should know it's FeCl3, not zinc, not reagents A and B, not hydrogen peroxide, not Kovacs reagent, not methyl red, not 1-nasol, all those other reagents. Know where the reagents are used and what the results mean for the test that they're used for. Is that clear? That's another thing to be aware of. Okay, because we're almost to the end. Let's talk a little bit about bacteria and the cells and the shapes. You know that stuff's coming back too. How would you classify this organism's characteristic arrangement, especially where the arrow is pointing on this one? So it's got a couple of mixtures on here, but it's got one big overall characteristic arrangement. This is a thing that has confused a lot of people on the test. You might see a couple of them stuck together in a line, but overall, if it's clumped together in clumps, that would be staphylo. Overall, if it's in chains, that would be strepto. So this was strepto. We saw staphylo on the exam. Are we clear on what the difference is between the two? Okay. What is the name and shape of the bacterial cells pictured? The name of the shape is the better. They look rod-shaped to me. What's the term for rod-shaped bacteria? And I'm not asking for the arrangement. I'm asking for the shape of the cells. So don't let that trip you up either. The shape of the cell, not the arrangement. So this is bacillus. So these are rod-shaped. Bacilli are rods. What's the word for round-shaped or spherical? Cocci. Coccus. Coccus for singular, cocci for multiple, and I'm not going to get picky about that. All right, so shapes and arrangement. Shapes, the only shapes that you're going to see on the test are going to be coccus or bacillus. The only arrangements that you can see on the test will be staphylo or strepto. If I'm asking about arrangement, they're going to have an arrangement. I'm not going to put no arrangement. I hate that. Okay? I want it to be clear because there's a lot to cover. So I'm not trying to make any of this confusing. If I'm asking about shape, I'm asking about shape, not arrangement. If I'm asking about arrangement, I'm asking about arrangement, not shape. And it might say grouping. That means the same thing as arrangement, okay? Remember that simple stains are a thing. If I simple stain something with methylene blue, I don't care if it's gram positive, gram negative, I don't care if it's a fungi, I don't care if it's a eukaryote. If I'm doing a simple stain with methylene blue, what color will it be? Blue. Okay. All right, let's go over the microscope parts. If you observe a specimen at 100x through a 10x lens, what is the magnification? We've got a 100x on our objective lens and a 10x ocular lens. What's the total magnification? What this really means is whatever's on your slide, you're looking at it 1,000 times larger than real life with your eyeballs. So this is 1,000 times. You want to multiply them together. Remember, when you multiply by 10, you just add 1 0. All righty, which lens do we use with oil immersion? You know this one. Easy question. On our microscopes, we use the 100x with oil. Know your parts of the microscope. Which part of the microscope generates the real image? The objective lens. It's the objective, I think this, right? So you're thinking this, yes. The twisty ones, yes. So the objective lenses generate the real image. The virtual image is the ocular lens. What's the piece that rotates the objective lens is called? That rubber piece at the top? It's the nose piece, okay? So maybe be aware of those, okay? All right, we have to be aware of lab safety. You will wear your lab coats. You will wear closed-toed shoes, for sure. Yeah, don't risk it. If you have to borrow a lab coat, you're getting points off for it, okay? So be aware of that. So I will have one and I'll let you take the test. If for some weird freak accident, everybody showed up without lab coats, I would let people borrow the lab coats and let them take the test. And if you still didn't have one, then I would probably let you still take the test in a testing center at a 25% reduction. So don't do it, right? So don't do it. I don't know why that would happen, but yes. So borrowing a lab coat will just cost you 2 points. But if you don't have a lab coat and I have to give you the test in the testing center, because of you not having a lab coat, it's a 25% reduction. Okay. To me, that's just, come on, at this point in the class. All I ask is lab coat and closed-toed shoes. I haven't made you wear long pants. I haven't made you wear goggles or gloves or anything. So I'm not asking much. Stains. Simple stain, gram stain, acid fast stain, capsule stain, endospore stain. All of the tests, the physiological, the selective, differential, enrich, those things, as well as the biochemical test, and all of that stuff. Be able to identify an organism. If I give you one of the charts, the chart that we had to fill out for the unknown report, that first page where you use positives and negatives and whatever, I'm going to give you a page that looks like that. In fact, I have it ready to make the test right now. I'm not going to let you look at it up close ahead of time, but it will be a chart that looks like this. And it's got results for gram positives and gram negatives. It won't help you that much to look at this because the chart that I'm giving is this page, okay? And it's all filled out. And I will give this to you, and I will give you a key. And it is different than the one that we have in class, okay? But you'll have to know how to use it, and that's the reference, okay? So it's set up the exact same way. So again, you'll start at the side, ask the question, is it gas or no gas in glucose? Is it gas or no gas in lactose? Is it, indole or positive or negative? And then find the organism and tell me what it is. Make sense? That sort of stuff. And you'll do that for a chart that I give you with a key, as well as a paragraph listing results in the key. I'll give you the key. I'm giving you the results. You just have to know how to use it. 2 questions on the exam like that. One with a chart, one with a paragraph. Do we have any questions about that? We're ready then, I guess. You all's just ready. All right, so that's the end of the review. Here's our little podium. If this were the exam today, And that, some of us wish that it was, because this was not bad, right? This was pretty straightforward. But let's see what our score would be. So good job today, guys. Thank you for showing up and doing your review. I think most people were here today. So good job with that. Our results for today, if this had been our exam, would have been a 67 average, which wouldn't be bad. But I'm hoping we'll do better. Now that we've reviewed everything and all that. So we're going to study up. We're going to do well on the lab final. And I'll see you guys on Monday. This time, lab coats and closed-toed shoes and phones and pockets.
On Monday, are you on what?
Yes. So on Monday, I will be clear. We do have presentations in class on Monday. It is still like we'll have a lecture activity where you fill out the same sheets. It's the same thing as we did on this last Monday. So it's the same thing. We will have presenters. Please show up and support the presenters. And then we will immediately follow that with the lab final. Any other questions?
Did you send the lab final monster?
Yes, the lab final will start at 11. Even if we finish lecture early, you'll have time to like just review or whatever after that. So we'll probably finish lecture early, honestly. We did last time. So we won't start lab final early just in case there weren't people there in person. And it'll give you time to, do your mini review beforehand. So 11 a.m. for the lab final. Okay, we good? All right, guys, have a great weekend. Rest up, study up, get your sheets turned in by midnight tonight. Yeah, we're almost there. You guys can go. Or if you want to see your Unit 3 exam, you can come look at that too.
Thank you, Professor.
Yeah, let me stop it. Thanks, guys.